Thank you for this opportunity to talk about these new compounds. So my disclosures are textbook royalties from Springer and Tima, not pertinent to the talk today. So I love this picture. This is the back of a chair in Independence Hall in Philadelphia, and it's the chair where George Washington sat when he was writing the Declaration of Independence. And Ben Franklin is quoted to have been commenting on whether he couldn't tell whether the sun was rising or setting, but as things progressed, he realized that the sun was truly rising. And so I think this is pertinent to this. This is such an exciting time in medicine, and as surgeons, perhaps we need to be worried that we'll be replaced by medical therapies. So Paul Ehrlich in the early 1900s proposed the idea of magic bullets. And the idea was to create compounds targeting specific disease-causing organisms and then to deliver a toxin to the organism using the compound. He actually received a Nobel Prize for this in 1908. And monoclonal antibody therapy, I think, is a part of his dream come true. So a little more history in the 1970s, it became understood that the B cells in multiple myeloma were all produced by a single type of antibody. And this led to an intensive study of antibody structure. In 1975, Kohler and Milson fused multiple myeloma cell lines with B cells, and they created hybridomas that could produce antibodies specific to known antigens and were immortal. And this won a Nobel Prize in 1984. In 1988, Winter and his team pioneered humanized monoclonal antibodies, and this eliminated the immune reactions that occurred in some patients. So just as a refresher, and really the purpose of my talk is to just set the stage for the more pertinent speakers to come, but what is a monoclonal antibody? So antibodies made by identical immune cells that are clones of a unique parent cell. They have monovalent affinity for a specific isotope, which, if you can't remember from medical school, is the part of the antigen that is recognized by the antibody. Monoclonal antibodies can be developed against antigens on cell surfaces, and two types of mechanisms can occur. Blockage of a cell surface receptor can disable the signaling pathway that would result from the natural ligand binding to the receptor, or it could create an antigen-antibody complex on the cell surface, which can trigger an immune response or result in cell death. It's possible to produce monoclonal antibodies to many substances, and there are many uses today. These are just tools in our sandbox. So we know about immunohistochemistry when we do our pathology, also Western blots. A lot of techniques use monoclonal antibodies. In analytical and chemical uses, you can purify compounds this way. You can perform immunoprecipitation. And therapeutically, we use monoclonal antibodies to treat cancers and inflammatory disorders. So what is a small molecule inhibitor? It's a low molecular weight substance that actually enters cells to affect other molecules such as proteins. It may result in altered cell function or cell death. And it's different from a monoclonal antibody. A monoclonal antibody is large and it does not enter the cell. It works on the cell surface. The advantages of these small molecule inhibitors are that they are low molecular weight, they can enter the cells, they can cross blood-eye and blood-brain barriers, they are rapid inactivation of their targets, and they're highly penetrant. They can allow researchers to study and control many aspects of cell biology, including cell cycle control, signaling, and gene expression. So the nomenclature was always a mystery to me until I put this talk together. So hopefully this will help to clarify. So really basic, if it's a monoclonal antibody, MAB, so it ends in MAB. If it's a small molecule inhibitor, IB, so that's your ending. The specific name of the monoclonal antibody describes how it was made and what the target molecule is. So there's kind of three parts to the names. So the source of the antibody is indicated by the suffix. So murine is OMAB, chimeric human mouse is ZMAB, humanized mouse is ZUMAB, and fully human is UMAB. And here's just another way of looking at this to put it in your brain. So there's different percentages of murine versus fully human, and then they each have a little bit different name. And of course, the more fully human they are, there's less potential for immunogenicity. The target of the antibody is indicated by the insertion. So the circulatory system, it's going to have a CI or CIR in the middle. Immune system, it's going to have an LI or LIM in the middle. And tumor is going to have a T or TU in the middle. And the name of the small molecule inhibitor describes the target metabolic or enzymatic step. And so you can see those here. So traditional treatments for cancer and immune disorders are pretty nonspecific. They affect all cells with high mitotic rates, and they can result in significant side effects. But molecularly targeted agents can specifically target cancer cells or inflammatory mediators with little or no effect on normal body tissues. And so they have really completely changed how we treat many disorders for which we found targets. It's a huge change in the management of diseases encountered in ophthalmology and oculoplastics. Thank you.